Okay, I'd like to thank the organizers for giving me this opportunity to speak. It's a real pleasure to speak to you today. Actually, the last time I spoke in this room was Recom 2009, where I described work from my PhD lab developing the sort-seq MPRA. And there we were using MPRA techniques to study to develop quantitative biophysical models of transcriptional regulation in bacteria. The work I'm going to tell you about today, which my lab did jointly with Adrian Craner at Cold Spring Harbor Laboratory, develops quantitative biophysical models to describe how splice-modifying drugs are able to treat human genetic disease. So it's a real pleasure to be able to present the story from the same podium. So, forward. Okay, so my lab studies the biophysical mechanisms of gene regulation. We do this by quantitatively measuring and modeling sequence-function relationships. Our experimental work uses massively parallel reporter assays to study gene regulatory sequences in two biological contexts. Like I said, alternative mRNA splicing in humans and transcriptional regulation in bacteria. Our computational work develops algorithms and software for analyzing MPRAs, but also other MAVE experiments. It increasingly focuses not only on biophysically understanding MAVEs, but biophysically understanding genomic deep neural networks. And our mathematical work addresses fundamental questions that arise in the process of this computational work regarding both the inference of sequence-function relationships from high-throughput data, but also the quantitative nature of sequence-function relationships more generally. So spinal muscular atrophy is a devastating genetic disease. It's caused by insufficient SMN protein. The disease is the most common monogenic cause of death in infants, affecting about 1 in 10,000 live births. SMA is caused by homozygous loss-of-function mutations in the gene SMN1, which codes for the ubiquitously expressed and essential protein SMN. So this talk is going to focus on two drugs, risdiplam and branaplam, which have been recently developed for treating spinal muscular atrophy. The way these drugs work is they restore the inclusion of exon 7 in the very close paralog so in this gene SMN2, which is a very close paralog of SMN1. It codes for exactly the same protein as SMN1 does, but it has a C to T mutation in exon 7, in position 6 of exon 7. And because of that, exon 7 gets skipped in about 90% of endogenous transcripts, and that skipping causes the adding of a degron tag to SMN protein, making it unstable. In the presence of risdiplam or branaplam, however, the inclusion of exon 7 is restored, and thus you get fully functional, stable SMN protein. So risdiplam and branaplam both work, at least in part, by stabilizing the binding of the U1 SNRP to the 5' splice site of SMN2 exon 7. In the absence of drug, U1 does bind the 5' splice site, but it does it somewhat weakly. This is a non-canonical 5' splice site. And as a result, you get low exon 7 inclusion. In the presence of risdiplam or branaplam, however, those drugs actually bind to the U1 5' splice site complex, stabilize that complex, and thereby increase the inclusion of exon 7. Now, there are two key mysteries surrounding these drugs. Both were found by kind of blind screens. So they weren't designed to do a specific thing. So one question is, how are these small molecule drugs so specific for SMN2 exon 7, considering they're so small, but also branaplam is like substantially less specific for exon 7 of SMN2 than risdiplam is. So what is the sequence specificity of these drugs, and why is branaplam less specific than risdiplam? So structural data from Frederick Lane's group has suggested this bulge repair mechanism. So the U1, the 5' end of U1 forms an RNA double helix with the 5' splice site of exon 7. This mismatching causes a bulged adenine at position -1. What they found is that SMNC5, an analog of risdiplam, actually is able to bind across the RNA double helix, and a carbonyl moiety on the drug is able to form a hydrogen bond with the amino group of the -1 adenine, pulling it out of the minor groove and into the helical stack, thereby stabilizing the complex. But it seems like both drugs do that. So why is risdiplam more specific? Well, the prevailing hypothesis when we started this project was that risdiplam actually binds a second site within SMN2 exon 7, a purine tract shown here. And presumably risdiplam, this has been suggested by two different groups. Both groups suggested that risdiplam stabilizes a protein RNA complex, and that by doing so, the co-occurrence of these two drug molecules synergistically act to promote exon inclusion. So going in, we had these questions. What are the 5' splice site specificities of risdiplam and branaplam? Do these specificities help explain why risdiplam is more specific for SMN exon 2 than branaplam? And how important is the putative second binding site of risdiplam for the drug effect at SMN2 exon 7? The approach that we use is we collected data using massively parallel splicing assays, RNA-seq experiments, and precision dose response curves. And based on these data, we developed quantitative mechanistic models that describe the sequence-dependent effects of risdiplam and branaplam on splicing, and the concentration-dependent effects of these and other splice-modifying drugs. We have a number of results. I really want to focus, though, on these three because of time. So we showed, first, that the results actually falsify the two-site hypothesis for risdiplam activity at SMN2 exon 7. There is no second functional site. Instead, the results suggest a different model that explains why branaplam is less specific. It suggests that branaplam actually recognizes 5' splice sites in two distinct biophysical interaction modes. One is an interaction mode with sequence specificity very similar to branaplam, and the other is an interaction mode with specificity distinct from branaplam. So the hypothesis is that branaplam is binding all the 5' splice sites that risdiplam is, plus another set caused by this distinct specificity. And we also show that the quantitative specificities of these drugs, they're largely consistent with the bulge repair mechanism, but that both drugs actually recognize nucleotides, somewhat distal from the 5' from the bulge, and that those are important determinants of drug specificity. So now I'm going to show you the evidence, just some of the evidence for these conclusions, and I'm going to do it quickly. Quantitative. Okay, so in order to assess the two-site hypothesis, we just made mutations into this purine tract, this putative site. And what we found is that this is a general splicing activator, but it's a this purine tract, but it's a splicing activator that activates splicing independent of drug. If you delete it, the inclusion-exclusion ratio of exon 7 decreases by about 100-fold, but not in a way that's really drug-dependent. To study the actual 5' splice site specificities of these two drugs, we, like I said, we performed a massively parallel splicing assay. We tested all single and double mutations to this 5' splice site on SMN2 mini genes. Each variant 5' splice site was linked to about 1,000 barcodes. We transfected these into HeLa cells, treated without drug, with risdiplam or with branaplam, and then did isoform-specific sequencing of barcodes in order to quantify the inclusion-exclusion ratios. And what we found was interesting. So for risdiplam, we were able to find a qualitative IUPAC motif that very cleanly distinguishes 5' splice sites that are activated by risdiplam from 5' splice sites that are not activated by risdiplam. However, there is no analogous IUPAC motif that describes branaplam specificity. But there is an IUPAC motif that describes which 5' splice sites are activated substantially more by branaplam than by risdiplam. So we call this the risdiplam IUPAC motif, and this the hyperactivation IUPAC motif. This two-motif observation was also consistent with genome-wide drug effect values that we measured by biophysically modeling RNA-seq data. What we found was that basically all the 5' splice sites activated by risdiplam matched the risdiplam IUPAC motif, but the 5' splice sites activated by branaplam matched either the risdiplam IUPAC motif or this hyperactivation motif, or some of them matched both. So in order to formalize this hypothesis, we developed a quantitative biophysical model that describes the thermodynamic binding of the drugs risdiplam in one interaction mode with the 5' splice site U1 complex and branaplam in two different interaction modes. The risdiplam interaction mode had this sequence-dependent binding energy. The y-axis here is the Gibbs free energy in kcal per mole effects of each variant nucleotide at each position. Branaplam, and the model also describes a second binding mode for branaplam that's described by this hyperactivation motif. Now, what we found is that this model, in which risdiplam binds in one interaction mode and branaplam binds in two interaction modes, it fits the data far better than a model in which risdiplam and branaplam just each bind in their own interaction mode, even though those two models have the same number of parameters. So what we find in these two energy motifs is that both are consistent with the Bulger pair mechanism. They have an obligate adenine at position -1 and a stabilizing guanine at position -2. However, the risdiplam energy motif has an obligate adenine at position -4, as well as a very deleterious cytosine at position +4. The hyperactivation energy motif has an obligate adenine at position -3, but no real dependence on -4 or +4. So we conclude that the quantitative energy motifs of risdiplam and of the risdiplam interaction mode and the hyperactivation interaction mode, they're both consistent with the IUPAC motifs, and they're largely consistent with the Bulger pair mechanism, but less so, but there's other stuff going on there. So to sum up, the results clarify the mechanism of an FDA-approved therapy, risdiplam, and we hope will facilitate the development of new splice-modifying small molecules. The branaplam two-interaction mode model explains why risdiplam is more specific for SMN2 exon 7. And finally, just quantitative mechanistic modeling turned out to be a very useful tool for understanding the genomic specificity and molecular mechanisms of splice-modifying drugs. And finally, I'd like to thank Yuma Ishigami and Mandy Wong, the two postdocs who led this work, and my close collaborator, Adrian Craner, who did this project jointly with my lab. Thanks.
Super cool. I don't know if these drugs have been studied enough that we know something about their side effects, and if we do the off-target splice sites hit by these different drugs? So, and I want to just say branaplam and risdiplam because I just want to say those words because those are so fun. But are there any off targets that can explain side effects?
There are off targets. So splicing of FOXM1 is a major off target, and this is a really important point. So branaplam is no longer being pursued to treat SMA in part because it has greater side effects. But one of those off-target effects is actually a pseudoexon in Huntington. And because of that, branaplam is being pursued as a treatment for Huntington's disease, which has no FDA-approved treatments. However, the side effects are substantial, and it's not clear that there's a therapeutic window. What our results suggest is that that hyperactivation interaction mode, that is the interaction mode that recognizes the pseudoexon in the Huntington gene. So if you can do medicinal chemistry to eliminate the risdiplam interaction mode, you might be able to get a derivative of branaplam that actually specifically targets the Huntington much more specifically and has reduced side effects.
Any other questions out there? I mean, I guess one question is, what made somebody think this would work? That's so cool.
I have no idea. These splice-modifying small molecules are bizarre. You know, there are 200,000 to 300,000 exons in the human genome, and they'll target like 100 of them. And they're tiny.
Right.
So it's a big mystery. These are one main class of splice-modifying small molecule. The other is this kinetin family, which is being pursued for treating familial dysautonomia. And we're trying to study how that works next. But the mechanism for how that works is very unclear.
Okay, cool. Thank you very much.